Hi, welcome to Mitocast. I'm Daniel Levine, host of the series focused on scientists, drug developers, and others advancing an understanding of the role that mitochondria play in health and disease. And today I'm pleased to welcome Doug Wallace to the show. Doug is the director of the Center for Mitochondrial and Epigenetic Medicine at Children's Hospital of Philadelphia Research Institute. He's a pioneer in the field of mitochondria biology, and his work has been essential in elucidating the role that mitochondrial DNA plays in human biology and pushed our understanding of how it can drive diseases of aging, including heart disease, diabetes, cancer, and neurodegenerative conditions. We're going to talk about how he came to study mitochondria, some of the key highlights of his research, and how our growing understanding of mitochondria can lead to new ways to prevent or treat a large number of diseases. With that, let's welcome Doug to the show. As people think about mitochondria, they may think in terms of biology or even chemistry, but your interest in physics is what led you to the study of mitochondria. How did you first become interested in it, and what inspired you to pursue the field as a field of research?
Well, Dan, I've always been interested in three basic questions. Who am I? Where did I come from? And why do I feel bad? And those basic questions led me to decide that theology wasn't working, although I tried that, and that it would be maybe better to look at science. So I had the opportunity to go to Cornell because I was a New York State resident, which was great. And I went there to learn everything I could about life, the universe, and everything. And got particularly excited about not only studying biology, but physics and chemistry, and trying to put the whole story together into some kind of meaningful understanding of where we are and why we're here. And that then led to a realization that biology is not just about the structure and the anatomy of animals, but in fact, the energy flow through the biosystem and through the individuals, and that's what animates us. And so I got very interested not only in physics because physics is interesting, but because of the physics of biology, which I thought was being very understudied. So I got the opportunity to go to Yale, and I was very interested as an undergraduate at Cornell about the work of a guy named Harold Borowitz, who was a professor at Yale. And he was a physicist, and he was interested in how energy flowed through biology. And he wrote a very nice book called Energy for Biologists, which I thought as an undergraduate was really cool. So after I got out of the service, I applied for Yale, got in. And so I had the great opportunity of spending one day a week doing theory with Harold Borowitz and 6 days a week working in Jerry Eisenstath's lab to try and figure out whether the mitochondria had a basically influence on our health and disease.
I could not possibly do justice to all the work you've done and the essential nature of it, but could you talk a little bit about the key discoveries you've made in the realm of mitochondrial DNA research and how these have shaped our understanding of genetics and human health?
I got to Yale, and I was in the Department of Human Genetics as well as microbiology. But I was interested in human genetics, but I came to realize that most of the way medicine looked at human biology was from an anatomical point of view. And that comes back to a guy named Vesalius that lived half a millennia ago, and he defined the structure of the human body. And ever since then, people that get trained in medicine are taught first about anatomy. And the first thing they asked to do, of course, is to dissect a cadaver. And I always tell the medical students, well, it's all great that you now know all the anatomy of the cadaver, but there's one problem, it's dead. And if your patient leaves your office in the same state as the cadaver, you've made a big mistake. So I think we need to know something fundamental, which is the difference between being alive and dead. And so that then led me to figure out, well, what is the energy flow that might be relevant, and why haven't we studied it? So what has been really a mainstay of human genetics for the last 150 years goes back to a man named Mendel and he worked on pea plants, and he noticed that certain traits were inherited equally from mother and father. And he proposed that there were two factors. He didn't know the word gene at the time, but we'll say genes. One coming from the mother's egg and one from the father's sperm, and they made a zygote which had two copies, and that then gave rise to the individual. And in the beginning of the 20th century, many Drosophila geneticists were studying the anatomy of fruit flies, and they then realized that most of the anatomy genes followed this Mendelian inheritance. And so, in fact, almost everything in genetics at the beginning of 1970 was about Mendelian genetics. But I got interested in the fact that the energy was so critical to being alive, and that the organelle in the cell that made most of the energy was the mitochondria. So I figured that studying the mitochondria couldn't be trivial. And inside the mitochondria, it had just been discovered that there was this closed circular DNA called the mitochondrial DNA, which we could isolate. So the first thing that I set out to do was to determine if that mitochondrial DNA had any genetic components. So working in Jerry Eisenstadt's lab, as I said, we were able to isolate cells that were resistant to a chemical called chloramphenicol. And chloramphenicol inhibits the protein synthesis of the mitochondria. And I must digress that our cell is made of two life forms, a nuclear cytosolic life form that came from an archaeobacteria and an oxidative bacteria that gave rise to the mitochondria. So both of these life forms have their own DNA. DNA goes into RNA, RNA goes into protein. And so, in fact, the chloramphenicol resistance was inhibiting the ribosomes, the protein synthesis of the mitochondria, but not of the cytosol. So the first thing to determine was chloramphenicol resistant due to a mitochondrial DNA rather than a nuclear mutation. And so what I invented was I took cells that were chloramphenicol resistant and I removed their nucleus, and I fused the chloramphenicol resistant cytoplasm with this mitochondria to a chloramphenicol sensitive cell, and I was able to show that I could transfer chloramphenicol resistance from one cell to the next, meaning that it must be in the mitochondrial DNA. So that was the first real event that led to understanding the genetics of this unique DNA.
How has the mitochondrial genetics field advanced since you began your research, and have there been technological advancements that have been pivotal in driving progress there?
Absolutely. So the first thing we had to do was figure out, using these cultured cell systems, how the mitochondrial DNA behaved. And we realized that there were hundreds to thousands of copies of this mitochondrial DNA in every cell. And so you could have a mixture of mutant and normal mitochondrial DNAs, which we then called heteroplasmy. And we also learned that that heteroplasmy could segregate as the cells went through cell growth, division, and you could get different percentages of mutant and normal. And the more mutant you had, the less energy there was, and that then gave the cell its disease phenotype. So then with that system, we began to look at then the human system. And what we found is that we could begin to study human mitochondrial DNA using a newly discovered enzyme called a restriction endonuclease. And this was discovered in the mid-1990s. And these enzymes see a particular sequence in a DNA and cut it into pieces. And so the sequence of the DNA will determine how many pieces that a particular enzyme cuts. So I got access to one of these restriction enzymes before actually they were even commercially available. And I had developed a way to isolate mitochondrial DNA and convert it into a radioactive probe using E. coli RNA polymerase and P32. And then I could take this restriction enzyme, and I had cell lines from families, digested their DNA with the restriction enzyme that gave different patterns for different mitochondrial DNAs. And then there was a new method from a fellow named Southern that had not been published, but we got a preprint of that paper. And there you can take the fragments of the DNA and run it on a gel, which separates the fragments by size, transfer it onto a paper, and then hybridize it with your radioactive probe. And when I did that, I found that the mother and the father had different mitochondrial DNA patterns, but all of the children had the same pattern as the mother. So that then proved that mitochondrial DNA was uniquely maternally inherited. And that's then a really important advance because everybody was looking for inheritance coming equally from the mother and father, but so they completely overlooked this DNA that was coming only from the mother. And what we then went on to find is that the most important energy genes are in the mitochondrial DNA. So everybody was absolutely missing energy genetics because they were never looking at the mitochondrial DNA because they didn't have the right inheritance pattern. So that was, I think, then the next stage that set us to looking for disease.
How well understood is the significance of mitochondrial DNA in relation to nuclear DNA and how they interact to influence various aspects of human health and disease?
So when the two organisms came together about 2.5 billion years ago, they were both bacteria. They had the same DNA, RNA, protein structures as any other bacterium. But what's interesting about bacteria is that they can exchange genetic information. And so it turns out that the oxidative bacteria that gave rise to the mitochondria transported most of its genes into the DNA of the archaeobacteria, and that gave rise to the chromosomes. So now the mitochondrial genome involves about 1,000 plus genes in the nucleus, plus the mitochondrial DNA, which basically is the wiring diagram of the power plant. So the way that Mother Nature set this up is just like you would build a power plant there in California. You would have all of the structure of the power plant with the city manager, but the actual wiring diagram of each power plant has to be in the power plant itself. And that's exactly how our mitochondria are set up. So you and I have very different wiring diagrams for our mitochondria because we have slightly different energy metabolism. And the reason maternal inheritance is so important is we wouldn't want to mix the different components of yours and my wiring diagram because yours may be more efficient in one way and mine in another. And if we put them together, we'd short out the circuit, and that would be a problem. So Mother Nature realized that there was a big problem, and the problem, of course, was men. So what she decided to do is just eliminate men and have maternal inheritance, and then there'll be no recombination, and yours and my mitochondrial DNA could never be mixed. And so that, I guess, is good for you and your mother.
What insights have we been able to gain from studying mitochondrial DNA variations, particularly in relations to complex diseases like autism or diabetes or Alzheimer's and cancer?
There are three things that we now know about the changes in the mitochondrial DNA that give rise to disease. The first is you could have mutations in the mother's mitochondrial DNA, and if they're deleterious, they can give rise to a maternally inherited disease. And we now have 100 well-defined mitochondrial DNA diseases and up to 1,000 potential mitochondrial DNA diseases. So this is a very high level of mutation for a very relatively small piece of DNA. The second thing is that because the mitochondrial DNA is maternally inherited, it's been transmitted from mother to daughter throughout all of human history, all the way back to what the Los Angeles Times called mitochondrial Eve. And mitochondrial Eve was the beginning, the first mitochondrial DNA in humans. And then that mitochondrial DNA has been transmitted from mother to daughter throughout human history. And as women migrated around the world, they came into different areas on the planet which had different environments. So like in Africa, if you want to have mitochondrial DNA that allows you to survive, you want to have a mitochondrial DNA that makes the maximum amount of energy, called ATP, with the minimum amount of heat, because every calorie you burn is a unit of heat. So you have very efficient mitochondrial DNA, and then you can run away from lions. But in the Arctic, the lions froze to death. So that's not a big problem for people. The problem for people was that they were freezing to death. So what happened is that the mutations in the mitochondrial DNA decreased the efficiency of making ATP, but then you had to burn more calories and made more heat. So that meant that they could then adapt to this cold environment. So in fact, every time our female ancestors moved into different environments, they adapted through their mitochondrial mutation to the unique environment that they have. which is why yours and mine and everybody listening to this podcast has a slightly different mitochondrial DNA, and why we can't mix these power plant wiring diagrams by recombination. And then the last thing, just a moment, the last thing is as our cells divide, they accumulate damage to the mitochondrial DNA. And as we get older, we accumulate more and more mitochondrial DNA damage. So when I was about in my 50s, I asked my mother what it was like to be aging. She was then 75, and she said, I just don't have the energy I used to. Now I'm 75, and I absolutely agree with her. In fact, we lose about 50% of our mitochondrial efficiency between the ages of 30 and 75. And that's because we've accumulated damage to the mitochondrial DNAs. And every time you knock out a mitochondrial DNA, you knock out a power plant, because without its wiring diagram, it doesn't work. So we start out with thousands of healthy mitochondria when we're children and have lots of energy, and just watch a child running around. It's really astonishing. But that by the time you get to my age, I'm hardly get up the stairs. And that's because probably I have a lot of mitochondrial DNA mutations that have accumulated over time. So those three kinds of mitochondrial variation then give the unique kind of genetics that nobody ever envisioned with Mendelian genetics, that gives us a totally new way of looking at the disease process.
Given what we know today, what are the most pressing questions that remain unanswered in the study of mitochondria, and what areas do you think warrant further exploration?
What we've been able to establish in the last, since we first identified mitochondrial DNA diseases and published it in 1988, which I guess is 35 years ago now, that there are very severe mitochondrial DNA diseases, and they affect children. And that's where a particular mutation in either a nuclear mitochondrial gene or the mitochondrial DNA is damaging enough that in and of itself, it will cause a clinical disease. We call those primary mitochondrial disease. And clearly here at CHOP, we're worried very much about children. So we see a very large number of children with these primary mitochondrial diseases. And they're quite devastating, and we're working hard to try and develop therapies for that. But equally as interesting is the whole effect of mitochondria on aging and age-related diseases like diabetes and autism and Alzheimer's disease and macular degeneration and hearing loss. So how could those diseases be a mitochondrial defect? Well, what happens is you get mild subclinical changes in the mitochondrial DNA. Let's say like a mutation in a gene in the nucleus called the APP gene that gives rise to what's called beta amyloid in Alzheimer's disease. But that mutation doesn't make you sick as a child. You don't become sick until much later in life. So why does it wait? If it was purely Mendelian, you would be sick when you inherited it. Well, what we now know is those genes actually cause mild mitochondrial defects. And then as we age, we accumulate damage to the mitochondrial DNAs in the cell, and they then augment the inherited subclinical effect until you have enough damage that you cross that clinical threshold and you get a neurological disease. And the reason that's so important is the brain is only 2% of your body weight, but uses 20% of all your cell's energy. So in fact, a very subtle change in mitochondrial energy will give you a very, very severe neurological disease. And so the reason we haven't understood these neuropsychiatric disorders like autism or Parkinson's or Alzheimer's or depression, so on, is because we've only looked at the nuclear encoded genes, and we haven't understood how they could have a delayed onset progressive course, which is that second component of the mitochondrial DNA, the age-related accumulation of the damage that then pushes you over the brain minimum energy threshold and gives you then the clinical problem. And that goes for heart disease, renal failure, liver's failure. All the organs that we have common diseases in are the most highly energetic organs of the body. And that's exactly why we believe they're mitochondrial diseases.
Well, considering the essential role mitochondria play in energy production and metabolism, how well would you say we understand the role in maintaining cellular function and overall health is understood?
In fact, this is a very new field. The first mitochondrial DNA pathogenic mutation was published in 1988 when we showed a particular kind of blindness was maternally inherited due to a mitochondrial DNA point mutation. So we didn't medicine didn't even know there was such a thing as mitochondrial disease until 1988. And of course, even now, it's not taught really in medical schools in any significant way. So this whole area of energy metabolism in health is really outside the purview of Western medicine. And one of the big challenges, and what I hope with MitoWorld and our colleague Gordon Freeman, is that we can bring this story out to more people to help them realize how important understanding not only anatomy but energy will be in understanding human health. And I really believe once we begin to look at the energy of disease, we will have a whole new repertoire of interventions to both prevent disease and also to treat it. And I think there's a huge opportunity here that only very few people around the world are actually exploring. And I think that's a great tragedy, because I think we could do a lot of good.
I know it's possible that there can be two or more mitochondrial DNA variants within the same cell. This is known as heteroplasmy. What are the implications of this for genetic inheritance susceptibility and the susceptibility to disease?
We have innumerable families where we have a maternally transmitted mitochondrial DNA mutation that is, as you say, heteroplasmic. Let's say that the woman's egg has a certain percentage, say 50% of a deleterious mutation and 50% of a normal mitochondrial DNA. Now, when that egg gets fertilized, the egg starts undergoing division, and the mitochondrial DNAs do not replicate until you get all the way into what we call the blastocyst, which many thousands of cells. So what's happened is those mitochondrial DNAs have been distributed into the cells more or less stochastically. So it's like if you had a big bottle of black and white balls and you reached in and grabbed only two, would you get two white balls, two black balls, or a black and a white ball? And we get that same kind of stochastic segregation in the female germline. So what we can get is we can have a woman who has perfectly normal body mitochondrial DNA, that is mostly normal mitochondrial DNA, but all of her eggs can have mostly mutant mitochondrial DNA, and all of her children will die of pediatric disease. So this segregation of heteroplasmy occurs both in our tissues as we develop, and also in the germline of the female lineages. And it makes it very difficult to do accurate genetic counseling because there's this fundamental stochastic component to mitochondrial genetics that doesn't exist in Mendelian genetics.
You've alluded to this growing body of research that implicates mitochondria in a wide range of conditions. What's the potential for developing therapies that target mitochondria to address things like metabolic disease and more importantly, maybe neurodegenerative disorders?
So there are really two ideas that we have to think about. One, for the late onset diseases, as I said, people often inherit a variant of their nuclear gene that decreases their energy metabolism a little bit, but is not bad enough to make them sick at that time. But then as they age, they accumulate these additional changes in their mitochondrial DNA, which augment that, and that ultimately causes their energetics to fall below what's necessary for that organ, and then you get a malfunction. It's sort of like a metropolitan brownout with more and more power plants going offline. So basically, from the common age-related diseases, we have two basic approaches. One is to intervene in the subclinical variants early in life, let's say change the APP gene to make it a more normal one, thus meaning that they don't start with a slightly lower energy capacity. But more useful, we believe, is actually decreasing the rate that we accumulate this mitochondrial DNA damage. And that second approach gives you then a universal way of offsetting the potential of an age-related disease for all the different kinds of diseases, the neurodegenerative diseases, the heart diseases, the renal diseases, because they all have a delayed onset and progressive course. And the idea is that all is the same mechanism that causes aging, the age-related decline in functional mitochondria, which results in energy deficiency until your organs are no longer able to sustain their normal function, and then you have a disease. So one of our main goals is to develop effective ways to protect the mitochondrial DNA from these age-related damage, and thus, in fact, offset any clinical problems, hopefully indefinitely, so that we can have optimal health for as long as possible.
Does that suggest anything about potential diagnostic tests that could identify conditions like Alzheimer's early in their progression before they manifest themselves?
So as we've got more information about the genetics of the mitochondria, the mitochondrial DNA, and the function of the mitochondrial DNA, which I'll add parenthetically based on another question you asked, the mitochondria have to communicate with the nuclear genes for the mitochondria because they have to work together. And it turns out that the mitochondria is a center for what we call metabolism, and many of the mitochondrial metabolites are actually factors that go to the nucleus and regulate nuclear gene expression. So the two organelles, the nucleus and the mitochondria, that are in constant communication and are working together to get an optimal health. So anyway, as we get more and more damage to the mitochondrial DNA, it also can send a signal to the nucleus and switch the state of the nuclear gene expression, and that the nucleus is trying to help, but actually it makes it worse. So there's another whole area of trying to intervene in this signaling system to make sure that the nucleus continues to do the most optimal energetics. So with that in mind, then, we have quite a range of potential therapeutic approaches that are being explored right now.
We talk about being at the start of an age of regenerative medicine. Most therapies at their best may halt progression of a condition, but is there any indication we might be able to reverse damage to mitochondria?
Yes, a number of us are very interested in whether we could actually fix the mitochondrial DNA. Let's back off, though. A number of the primary mitochondrial diseases, remember, are nuclear encoded genes. So those we have developed, that is we, the larger we of scientific community, is developing pretty good effective ways of introducing into cells healthy genes where there's damaged ones. So we might talk about adeno-associated viral gene therapy, or in fact, lipid nanoparticles to deliver genes or RNA to cells to correct nuclear encoded gene defects. That's much easier because there's only two nuclear coded copies. So we only have to fix one or the two of them to get the patient to be more healthy. The problem with the mitochondria is there are thousands of mitochondrial DNAs in the cell. So even if we could fix 1 or 5 or 10, that still doesn't cross the expression threshold to make you healthy. So there's a lot of effort then to think about how we could modify the mitochondrial DNA to cause the majority of the mitochondrial DNA to be in the good form. And so right now, one approach is that we call gene editing, in which you can actually put proteins into the mitochondria that could go and find a particular mutant mitochondrial base and substitute it. But they have to be pretty efficient. That is, you would have to do like 50 or 60% of the mitochondrial DNAs of every cell of your body would have to be changed or repaired to give a healthy outcome. Another approach that we're working on is trying to import nucleic acids in the mitochondria to actually fix the problem. And that is still research, but that would be more general, because then you could put in many copies and that they would all fix all the mitochondrial DNAs at the same time.
We're talking about fairly expensive modalities right now. Are there other challenges researchers face when attempting to develop treatments that target mitochondria? And are there other innovative approaches being explored?
Well, I think this is a good segue to talk about something that probably a lot of people have heard about, which is the so-called three-parent babies. That, of course, is now legal in Britain, and I think is becoming legal in other countries. And here the situation is if a woman, for instance, herself reasonably healthy, finds that all of her eggs are tainted with a high level of mutant mitochondrial DNA, then we know that she has a very high probability of all of her children being very sick and perhaps dying. So what people in Britain did in Newcastle is, and also in Oregon at the Oregon Health Science Center, they take the oocyte and remove the nucleus from the oocyte, so the nuclear genes are fine. Take a normal, healthy oocyte from a female donor, remove its nucleus, and put in the nucleus of the mother with the tainted mitochondrial DNA, and then fertilize that egg. And then now you have, in fact, the mother's and father's nuclear genes, but another lady's mitochondrial DNA. And so that approach then is one way innovatively to remove the problem of damaged mitochondrial DNA in the oocyte. But as you point out, that is a very laborious and expensive procedure. So we would love to find better ways to intervene. One way that, of course, we can do is we can use this pre-fertilization genetics to find out which eggs have high damaged mitochondrial DNAs and which don't, and then, in fact, maybe preferentially use those eggs. Or if we could, in fact, get mitochondrial editing, DNA editing, maybe we could actually edit the damaged mitochondrial DNAs and help in that way. But all of these are going to be very long-term, expensive procedures to develop. And a lot of people are interested in them, but we don't have them, of course, right now in our clinical repertoire.
And that last one you described, would that be considered mitochondrial replacement, or is that something else?
Where you have exchanged the nucleus for into a different egg, that has been called mitochondrial replacement therapy. That's correct. But that term can also be used, for instance, if you could actually get cells to take up mitochondria, and they could then augment the damaged mitochondria in the cells. And there's a growing excitement right now about, in fact, actually having mitochondria taken up by sick cells and helping their energy get better. And that also is called mitochondrial replacement therapy. So both are actually quite exciting at this point.
Are there any ethical considerations surrounding mitochondrial research, particularly concerning genetic manipulations and its implications for future generations?
Of course, there's a great concern about nuclear gene editing because those genes are transmitted and would be carried through the chromosomes indefinitely. The argument that has been made is that the mitochondrial DNA is a separate entity, and therefore you aren't damaging the mitochondrial DNA, you're simply changing it for another mitochondrial DNA. So these approaches are not actually altering the genome itself, they're changing the genome from one to another. And at least in Britain, that has been felt to be a different kind of category to the kind of chromosomal genetics that have been proposed. In the United States, of course, that still falls into the same issue, and it's not legal to do three-parent babies in the United States. But so there are definitely ethical issues for all of these discussions, and I certainly honor all the different opinions. But I will say that there's nothing more painful than to tell a mother who's had several miscarriages or devastated children that there's nothing we can do for her because she has a probability of 1 of transmitting her mitochondrial DNA, and that's very difficult.
We've certainly seen a broad movement in medicine towards personalized medicine and precision healthcare. Given that there's unique genetic characteristics to mitochondria, what's the potential impact on individualized treatment strategies?
Remember, I told you that as mitochondrial Eve gave rise to all the different mitochondrial lineages of women that went around the world, we now have a wide array of very interesting mitochondrial DNAs with different properties. They're more or less sensitive to cold. They're more or less efficient. They have different sensitivity to infections, to cancer. And these kinds of changes, of course, might be subject to the kind of mitochondrial DNA exchange that we've been talking about. But I think we need to be careful to think about that, because these same mitochondrial DNAs were good in one context, that is wherever they first appeared and came to predominate. They may only become slightly deleterious when they move into a different environment, say move from, change the caloric intake, let's say, of a Western European high-fat diet onto, say, a population that lived in a very limited environment, had much more efficient mitochondria, then the high-fat diet would overload the energetic system and result in diabetes and obesity. So we have to be cognizant of the fact that Mother Nature gave us all this diversity for a good reason. And I would rather honor the diversity than try and figure out ways to switch it around.
A lot of people come to the mitochondria through the study of other systems and diseases. How important is interdisciplinary collaboration in advancing our understanding of mitochondria's role in health and disease? And what fields do you believe are crucial for further exploration in this context?
It falls back on some of your earlier questions, which is, why isn't this idea more generally thought about and accepted in the medical community? And the reason for that, again, goes back to Vesalius and to this anatomical paradigm of disease. Basically, when you get into medical education, you're often urged to form a clinical subspecialty, that is, to become an ophthalmologist or neurologist or cardiologist. But that then results in a bias, because then you're assuming that if there's a problem with an organ, like you have a headache, then you need to go to the neurologist because there's something wrong with your head. But actually, what we've just talked about is that if you have a mild energy defect that's systemic throughout the whole body, it will give you a very bad headache. But there's nothing wrong anatomically with your head, but there is something wrong with your energy. So what's happened is that we have NIH institutes that are all organized around anatomy. And we have clinical departments all organized around anatomy. So as a result, we don't have a unifying view of how bioenergetics affects all the different health problems we have. So what we really need is to find ways to bring this community together. Now, one area that's been done in the primary mitochondrial disease is an organization like the United Mitochondrial Disease Foundation, which has worked hard to bring families together with clinicians to help the children. But we don't have that kind of structure for the common diseases. And right now, if we want to study, say, mitochondrial involvement in psychiatric disorders, we would go to the National Institutes of Mental Health. But their perspective is that you want to do nuclear gene analysis of genes that affect only the brain, when in fact, I think a lot of psychiatric disorders are systemic energy disorders. So this anatomical paradigm has limited our ability to communicate and work together to really advance this field. And I think that is an unfortunate factor, but maybe things like MitoWorld or other efforts that people are making can begin to broaden our perspective and maybe bring some of these international efforts together into a coherent forum.
You've had a long and remarkable career. What advice would you give to an aspiring researcher interested in pursuing a career in mitochondrial genetics based on your own experience and contributions?
I couldn't be more excited about the future of mitochondrial medicine. Again, I go back to the fact our cells are made of two separate life forms. We've spent almost all the money and effort on one of them, and we've left the other one we're almost completely ignorant about the mitochondria. We don't know how the genes are regulated. We don't know how they interact closely with the nucleus. We don't know a lot about whether they're edited, whether they're viruses that affect them. Almost an unimaginable biology that we are completely ignorant of. And every day we're getting exciting new discoveries by only the handful of people around the world that are working on this, but each one is more remarkable. One of the things that came out of Harvard, which I thought was amazing, if you have a cancer cell, the cancer cell will sidle up against a lymphocyte. the cancer cell is dividing and dividing, and its mitochondria becomes sick over time because it doesn't go through sexual reproduction. So what it does is it causes a tube to be drawn out of the lymphocyte, and it sucks the mitochondria out of the lymphocyte to reconstitute its own mitochondria. Now, I would have never imagined 20 years ago that that would be possible. I think there's a whole ecology of mitochondrial biology in humans and in plants that we have never even imagined. And I just think it's such an exciting field that I think if we can get the community to support it, will be a wonderful opportunity for young people. But we have to get beyond this idea that all of the problems are going to be solved by nuclear genetics and simply sequencing hundreds of thousands of nuclear genomes. As an example, it's been declared that we now have a complete understanding of the genetics of height. So that was done by what's called GWAS, so looking for genetic loci that correlate with height through hundreds of thousands of cases. But when you ask the GWAS geneticists, okay, what percentage of height determination can you now explain? It's less than 50%. So you say, well, what about the other 50%? Oh, that must be environmental. No, what that means is that they didn't study it in the nuclear genome. But that doesn't mean that it couldn't be mitochondrial DNA variation, which they never look at. So I think that there's just a huge opportunity to move a lot of what seems to be our blind spots into the light and to really make some really exciting discoveries.
Doug Wallace, Director of the Center for Mitochondrial and Epigenomic Medicine at Children's Hospital of Philadelphia and Professor of Pediatrics at the Perelman School of Medicine. Doug, thanks so much for your time today.
Danny, thank you for your interest and all the listeners for their attention. Remember, your mitochondria are your energy, and you have to thank your mother.